Hey, good morning and welcome everyone to our webinar. So I'm Eunice. I'm from Apical Scientific. Currently, I'm the salesperson who's handling for Summer University. So hope everyone had a great weekend and we're very happy to have all of you to join in today's session. So for today, we'll be bringing you the techniques of cell culture. So there will be 2 sessions in today's webinar. Whether you are a seasoned researcher or new to cell culture, hope this webinar will be a guide or refreshment for everyone. So we do have also some quizzes at the end of each session. So be sure to stay logged in as there's some interesting prizes to be won. So to introduce today, our speaker for today, Miss Mana from Nacalai Tesque. She has an extensive experience of 13 years in R&D and expertise in the field of biochemical. So feel free to drop some questions in the chat box. So we'll be sure to guide you out at the end of every session. So do also keep yourself on mute to ensure a good experience for everyone. Without further ado, Miss Mana, you can take over from here.
Yeah, sure. Sorry, wait, please. Okay, so can you see the slide? Is it okay? Okay. So, hi everyone. Once again, my name is Mana Shimotsuma. I had been R&D department followed by 3 years of experience in marketing in Japan. And now I belong to international business department. I have been developing product for more than 15 years, especially in the biochemical field. I'm so pleasure to talk with you guys. First of all, let me introduce our company, Nacalai Tesque. This picture was taken around our laboratory, Bamboo Grove in Japanese, Chikulin. It's not popular tourist spot, so it's usually quiet, but the atmosphere is kind of traditional Japan, Kyoto. It's really nice. Nacalai Tesque was found in 1846 in Kyoto, Japan. We have a long history. Now we have 16 sales offices, 1 laboratory, 1 factory, and 1 logistics center in Japan. Our product line by reagent such as cell culture medium or reagent for SDS page Western blotting or immunohistochemistry. About the HPLC column, our brand name is Cosmoshield. 4 years ago, we celebrated 40th anniversary and fine chemical and research chemical. By the way, he is our company's mascot, Bottle Tesque. During today's webinar, he sometimes will appear, so please check him. Okay, so thank you for listening about the introduction of our Nacalai Tesque. Let's move to today's webinar, Mastering Cell Culture. This is today's agenda. We have 2 parts, the basic knowledge and the practical handling. Each part we will have Q&A section, so please don't hesitate to ask us and write your question on the chat box during my explanation. Anything is okay. Any comment or question are welcome. Okay, so let's start from first section, the basic knowledge of cell type. Roughly speaking, we can classify cell type into 2 types, primary cells and cell line. Primary cells are isolated directly from human or animal tissue. On the other hand, cell lines have been continually passage over a long period of time. The difference point between primary cell and the cell line in brief, cell lines are easy to culture or maintain. On the other hand, primary cells are difficult. For example, in primary cells, special techniques for isolating cells from organisms are needed. And for maintain, originally additional nutrients are not included in basal medium are needed. In other words, for primary cells, you need special techniques and special medium. Moreover, primary cells have limited lifespan. But primary cells are representative of in vivo tissue compared to cell lines. Cell lines have lost the true characteristic of original tissue. About the way to get these cells, in the cell line, you can buy them from the supplier such as ATCC. In terms of primary cells, in some cases, you can buy primary cells from cell supplier, but most cases, you have to isolate them from tissue by yourself. Recently, iPS cells have been popular. iPS cell can differentiate into many different cell types found in the body. This property makes them valuable for studying development, modeling diseases, and potentially for regenerative medicine. Okay, so we move to next topics about the contamination. Regarding contamination, there are two types, cross-contamination and contamination with microorganisms. About the cross-contamination, cross-contamination is that your cells are contaminated with another cell unintentionally. For example, you originally culture HeLa cell, which is adherent cell, but there are a lot of suspension cells. In this case, you can doubt cross-contamination. To prevent cross-contamination, you shouldn't deal with more than 2 type cells at the same time. During waiting for centrifuge, you may want to passage another cell. If I be honest, I also think so, but we don't recommend it. About the contamination with microorganisms, this picture are example of contaminations. So do you know where contaminations are? With bacteria, here a very small one, and with fungus, this is a large size compared with cells and has branching like this. If contaminations are occurred in your cells, unfortunately, you should give them up. There are some ways to rescue cells which are contaminated, but it's hard and difficult work actually. For dealing with cells, aseptic techniques are essential. So let's review the basic operation for using a clean bench. You might think it is too basic, but I would say it is very important. So please let's review together. First, preparation. Before using the bench, turn on the UV light and clean the working area. Be aware that UV light is harmful to humans and cells, so make sure to turn off the light before starting your work. Then, run the circulating fan for about 5 minutes in advance. Next, check your cells. Key points are dramatic color changes in medium, contamination of foreign materials, and abnormal shape or proliferation rate of cells. If you notice any of these issues, you should discard the affected samples. Spray ethanol and clean working area. Also, instrument and reagent such as medium should be wiped by ethanol. Okay, so next, aseptic techniques. I would like to introduce several points for aseptic techniques. First, point 1. Control your working area in the clean bench. Keep the clean bench free of unnecessary items and bring only the essentials into the bench. Additionally, the airflow in front of a clean bench is easily disrupted. So use the center of the clean bench. And point 2, during operation, pay close attention to the pipette tip and the mouth of culture vessel to prevent contamination. Point 3, rigid bottles and the culture vessel should only have their lid open during operation. And operator's hand should not pass over containers with open lid. When placing dish over tube lid, either orientation of the lid, upside and downside, is acceptable. However, be aware that if the solution sticks to the inside of the lid, it may drip between the lid and the surface of the bench, which could lead to contamination. Point 4. If you spill culture media or reagent, wipe it up immediately with alcohol. Leaving the spill as it can lead to bacterial contamination. Point 5. Don't talk to operator during aseptic operations. It is important to prepare the environment and the physical condition so that you can concentrate on the operation. Okay, so last, cleaning. Bring instruments and reagent outside the clean bench. They may be contaminated by droplet of invisible culture media, so wipe them with alcohol to prevent carryover for next use. Wipe the working area surface and both sides of clean bench's front glass with alcohol. Run the circulating fan for at least 5 minutes after work, and then close the clean bench door and turn off the fan. Turn on the UV light. Since ultraviolet rays degrade plastic, it's best not to leave it on for too long. Turn it off after about 1 hour. And this is last point of basic knowledge, this section. I will talk about the basal medium. There are various basal mediums such as DMAM, RPMI, or HAMS F12. And these mediums are used depend on your cells. In most cases, when you buy the cells at the cell provider, you can also get the information to maintain the cells. Or another case, we refer to previous paper. Medium contains amino acid, vitamin, fatty, mineral, and glucose. There are a lot of different types within this medium. For example, about DMAM, we or other companies such as Gibuco or Sigma have several types of DMAM. Actually, we provide 10 types DMAM. It is so complicated. So I will share key points of the component of basal medium. First one is glucose. In most cases, there are 3 types: high, low, and without. You can choose them depending on your experiment. And about glutamine, with and without glutamine. Glutamine is easy to break down. That's why some researchers prepare the medium without glutamine and add them before use. Or another case, you can use a stable glutamine, L-alanine glutamine, or Glutamax. about sodium bicarbonate or HEPAs. These are buffers commonly used for maintaining the pH of cell culture. In case of sodium bicarbonate, you use them in presence from 4 to 10% carbon dioxide, normally 5% of CO2. CO2 in incubator and sodium bicarbonate in medium adjust the pH. Is it? It's okay? No problem? Okay, so medium adjust the pH of medium to proper pH. And about HEPAs, HEPAs has more buffering capacities than sodium bicarbonate. But please remember that the sodium bicarbonate is nutritionally necessary for most cells, so HEPAs should be added in addition to, not in place of sodium bicarbonate. And the last point, phenol red. Phenol red is pH indicator. If the color of medium with phenol red is purple red, the pH is alkaline. On the other hand, yellow, the pH is acid. We can know cell condition by the color of medium with phenol red, but phenol red interfere to absorbance measurement for cell-based assay like an MTT assay because of their color. And moreover, phenol red pretend as estrogen, which is one of hormone. So if your cells are sensitive for estrogen, such as MCF7, which is a human breast cancer, you should avoid using phenol red. So like this, there are many different type of DMAM with different composition. So please be careful when you order based on medium. Okay, so this is all for first section. And okay, so can you see the slide? Is it okay? So we move to next section, the practical handling. In this section, I will introduce the practical handling with videos. But first, I'm talking about the procedure of cell culture. As culture time goes on, the cell number increases. During proliferation, the cell takes up nutrients in medium like glucose, amino acid, or vitamin, and release metabolites. In the cell culture vessel, the nutrients are losing and the metabolites are filling. Finally, cell dies because of much metabolites and depletion of nutrients. Before this bad condition, cell has to be transferred to a new cell culture vessel with fresh medium, performing passaging cells. Or another case, cells in good condition are pulled up for backup of variable cells or because of long vacation. This section, I'm talking about cell detachment, cell count with trypan, and preservation cells. Let's start from passaging cells. There are two types of cells, suspension cells and adherent cells. For passaging cells, suspension cells require just cell count. Meanwhile, adherent cells require cell count and cell detachment. From next slide, I introduce the practical operation of cell detachment, adherent cell, HeLa cells. But before detaching the cells, please observe the cells and check the cell density. 70, 80, 90% of confluent is okay, but 100% of confluent is bad. The color of medium changed to yellow because of metabolites, and the shape of cells were distorted like this, and part of cells were floating. Before this bad condition, please perform passaging cells. Okay, so I'll show you the practical operation of cell detachment. At first, remove the medium using an aspirator. And the cells are washed by PBS. Add the PBS gently along the wall of cell culture vessel. And then remove the PBS. Next, add trypsin EDTA. For a 60 ml dish, 500 microliter of trypsin EDTA is sufficient. Incubate the cell culture vessel in 37-degree incubator. During incubation, cells change to round and get to float gradually. The incubation time is about 3 minutes. If needed, you can prolong the incubation time to 10 minutes. But we don't recommend to incubate for 10 minutes more because cells are damaged by trypsin. After incubation, please check the cells. The solution became muddy and thick, and you can see several clumps of cells. Add medium with FBS to deactivate trypsin. FBS contains trypsin inhibitors, so with FBS is important. If you culture the cells without FBS or serum, you use trypsin inhibitor, which are made from chicken egg or soybeans. Transfer the cells to new tube. And centrifuge the tube at around 100 degrees and 5 minutes. After centrifuging process, remove the supernatant. Add fresh medium. And pipette gently. So far, cell detachment is over. Go to next step, cell count with trypan blue. Next, I will show you the practical operation of cell count with trypan blue. Sort 100 microliter of the cell suspension to a new microtube. Please perform this operation in a clean bench. Add trypan blue solution and pipette gently. From this step, you can treat it outside the bench. And if you need, please dilute the cell suspension with PBS at this step. Inject the stained cell suspension between the counting chamber and the cover glass. Actually, 10 microliter of stained cell suspension is sufficient. Please count the cells using microscope and a counter like this. Please note, incubate the cells with the trypan blue for from 2 to 5 minutes. If not counted within this time, the cells will begin to damage and take up the dye. Counting chamber has 4 sections, 1, 2, 3, 4, and this is the image of one section of counting chamber with stained cells. Count the cell in this area. White cells like this are living cell, and black cells like this are dead cells. For cells located on line, like this, please determine which side are to be counted in advance. For example, you may count cells on the upper and right side line, but not bottom and left side. Count for section and calculate the average of number. For example, in this case, I mixed 100 microliter of the cell suspension and 100 microliter trypan blue solution. And that means this cell suspension was diluted by 2 times. And the cell number I counted was 95, 90, 87, and 88. So the average of counted cells is 90. So from dilution ratio and the number of cell count average, we can know that the concentration of this cell suspension is 180 times 10 to 4 cells per milliliter. This is the operation of cell count with trypan blue. I explained passaging cells. Actually, cell count step can be skipped for routine. In my case, suspension cells HL60 and the other hand cell HeLa, both of them are passaged at 5 times dilution, and 2 or 3 days after, I perform passaging cells again. In most cases, a good passaging ratio is written on the datasheet which cell supplier provide. So please refer the datasheet to get information on your cells. But before cell preservation, I will explain next, or cell-based assay, you need to count cells or check the viability of cells with trypan blue. Cells in good condition are passage, I explained already. Or another case, cells are preserved for backup of variable cells or because of long vacation. Next, I introduce how to preserve cells. We have medium for preservation, cell reservoir 1. Conventionally, cultured medium with FBS and 5 or 10% DMSO are used for preservation. Today's workshop, I introduced slow freezing method and recovery method with cell reservoir 1. The important points are the same for both the conventional preservation buffer and our product, cell reservoir 1. At first, I will explain about the slow freezing method. First of all, the day before freezing, verify the conference is between 70 to 80% via microscopy like this, and then replace the medium. It is important to ensure that cells are in the log phase of growth. We have to freeze wholesome cells. Dissociate cells using the standard procedure. And centrifuge it. Remove the supernatant. And resuspend the cell pellet in cell reservoir 1. The good cell density for cell freezing is at from 5 times 10 to 5 to 1 times 10 to 7 cells per milliliter. For conventional preservation medium, good cell density is the same. Dispense the cell suspension into cryopreservation tubes. In this case, 1 milliliter of cell suspension are dispensed. Immediately store these tubes at -80 degrees. To prevent cell damage, please perform from resuspending the cells with preservation buffer to freezing rapidly. For conventional preservation buffer or depend on your cells, program freezer like a BioCell are needed. Next, I explain cell recovery. At first, prepare 10 ml of fresh medium in a tube. The medium which is suitable for your cells are prepared. Remove the cryopreservation tubes from the freezer and immediately placed in a 37-degree water bath. To prevent the damage, not shake. Place the tube at water bath until being thawed partially. It takes about 1 or 2 minutes. And then put the tube in a bench immediately. Wrap the tube with ethanol. And add this cell suspension to the tube prepared. Centrifuge the cell suspension. After centrifuging process, remove the supernatant. Add 10 ml of new fresh medium. And resuspend the cell pellet. Transfer to new cell culture vessel. And then incubate the cells in an incubator. The key point of cell recovery step, osmotic pressure and cell toxicity of the MSO. After thawing, preservation buffer is not proper osmotic pressure for cells. And in case of with DMSO may show cell toxicity. So you should dilute the cell suspension after thawing with fresh medium rapidly. But sometimes there are cells which is very sensitive for changing of osmotic pressure. In this case, the defrosted cell suspension have to be diluted drop by drop. Okay, so explanation of preservation cell is over, and this is all for second session.